So the recording is on from this moment. So thank you everyone to be here for the population dynamics virtual meetings after the summer break. We have Susann Müller. Now I will share my screen and introduce the speaker. I hope you can see the first slide. So Susann Müller is from the Helmholtz Center in Leipzig in Germany. And the talk that she presents is about ecological forces that dictate microbial community assembly processes in bioreactor systems. So Susann got a PhD at the University of Halle in Germany. Then she became researcher at the University of Leipzig in the Faculty of Biosciences. And after this one, she became researcher at the Department of Environmental Microbiology at the UFZ Center for Environmental Research in Leipzig-Halle, and at the same time now is also professor for microbiology at the Leipzig University in the Faculty of Life Sciences. So she has several interests, one of which is the control of bacterial community growing in bioreactors. She's also interested in microbial community, in flow cytometry, and in general in population dynamics. And more broadly, also, she likes to study bio-based circular economy. So today, she will actually speak, as I told you, about ecological forces that dictate microbial community assembly processes in bioreactor systems. And with this one, I will stop sharing my screen. And the stage is actually yours, Susann. Please go on, and thank you for being here.
Okay, I'm sharing my screen. You see this full slide?
I can see the full screen, yes, works perfectly.
Full screen, yeah. Yeah, welcome everybody. So just let me see. Sorry, going back. Yeah, I'm back somehow out of it. Yeah, is this true?
I can see the full screen, but if you would like to have a pointer, I cannot see the pointer.
No, I'm just I don't see my slides anymore right now for the moment. So
Okay. So maybe you can try to reshare again if you prefer.
Sorry for this. Okay. So I want to talk about today about community assembly.
But we don't see full screen now. So we see your slides mode.
Oh, sorry. Why? My God, yeah, so sorry.
It's fine.
But
Still not full screen. Probably have to go to.
no?
No, we see still the presentation mode.
Because I'm sorry for that. Can I stop sharing here somehow? Because I have.
Yeah, I think you can actually stop. Maybe I can. Okay, now can share now, and now we can share again. Yeah. Now we can see full screen.
Okay. Also, before I start, I would just like to talk about a bit where I'm coming from. So I'm coming from the Helmholtz Center for Environmental Research, which was founded in 1991. And it's funded by the federal German government to 90%, and 5% are coming both from Free State of Saxony and State of Saxony-Anhalt. The main location is in Halle, but we have also locations in Leipzig, and we have also locations in Halle, Magdeburg, and Bad Lauchstädt. We have about 1,200 employees currently. I'm belonging to the research unit Sustainable Ecotechnologies, coming from the Department of Applied Microbial Ecology and working in the group flow cytometry as a group leader. So this is the background, and going on further. This is not working. Yeah, now. So we are working with microbial communities in different aspects. So we have a lot of corporations, and one is the cooperation with people from the medical sector. But mainly when you're going for communities from the medical sector, these people are working with sequencing data. This is about human health and the analysis of microbiomes with regard to variations between individuals, also with changes according to health status, age, gender, diet, and environmental conditions, and also about influences of the immune status of humans, and also the influence of the human microbiome on human metabolism and regulation, and many more. Originally, the microbiome research came from water research, actually, and especially when using flow cytometry, what we are doing in our lab, it's coming from surface water and ocean studies. Because these organisms there have autofluorescence, and it's quite easy or relatively easy to measure single cell signals in these samples, and they followed mainly the phytoplankton as which is the base of the food web, and which produce chlorophyll in several percent dyes, and so this is about how these microbes in the water phase driving biogeochemical cycles, and how the functional food web is established. But in our case, we are now more concentrating on bioreactor technologies, trying to understand how microbial communities can grow together in these confined environments. Because we have a lot of competition and cooperation in these reactors, we have influence to increasing product quantities by substrates, for instance. There are dependencies on metabolites or consecutive biochemical pathways. So there are lots of cell types, and this leads to, or can lead to, loss of connected metabolic pathways. And of course, what is most important, we have different growth rates. And we have all the positive things, of course, happening in these bioreactors, so shared metabolisms and intermediates, redundant functions, and interdependencies and support of microorganisms between each other. But bioreactors, I think, will be in the future part of the circular economy, because they can use waste material and produce, for instance, platform chemicals, which are necessary to replace chemicals that are normally produced by fossil resources. Nevertheless, using microbial organisms in biotechnology is still not really ongoing, besides the food industry, of course, but in the chemical industry. And the reason for this is that these communities are not structurally or functionally stable. And we want to contribute to understand why this instability is occurring, and if there are solutions to stabilize the compositional communities. Okay, and when people are investigating microbial communities, they need to handle the fact that most of these species that are inside of a community cannot be cultivated. And therefore, they need to use, to learn something about these communities, they need to use omics technologies. And this is, for instance, this metagenomics, metatranscriptomics, metaproteomics, and metabolomics approaches. But in our case, we want to use flow cytometry. And the reason is that compared to the omics technologies, we have quantitative information on cell numbers in the system, cell numbers per milliliter. The flow cytometry, besides that the devices are expensive, but as soon as you have one, the technology itself or the procedure itself is very cheap and it's very fast, and you have the data available within 2 to 3 hours, just ready to go for controlling bioprocesses. And this is something that we need. When you are going for the omics technologies, then you know all of those who are doing this know how long it takes to take the sample, to handle the sample, most of all to evaluate the data that are coming out of it. We also need to wait to get the data in, yeah. And then finally you have the results on the table, the reactor process is long run. And the aim for flow cytometry is that we get data in time to just have the ability to interfere with the process or to control it. Just to go on further for the method that we are using. So this is classical flow cytometry. We have cells running in the screen, and one after the other. This is hydrodynamic focusing, and then the laser hits the cell, and we can get information on the specific cell, on cell size. So it's related to cell size, not always identical to cell size like in microscopy, but it's related to cell size. And then we can get information on site scatter, which gives information on the density of the cell and the granularity. And then, dependent on if we have autofluorescence or if we have we stain certain parameters in the cell, we can measure additional fluorescent dyes in the cells. But in our case, we just use kind of this type of setting here. We measure only 3 parameters. So 2 of them are intrinsic, which is cell size and granularity. And we also measure DAPI content, meaning every cell has a DNA, and this DNA is stained by DAPI very specifically, and we can follow for each cell the amount of chromosome numbers per cell. And this, together with this forward scatter, gives the cell a certain position in the histogram. And each point, what we are seeing here in the histogram, is a cell, and it's in the z-axis, it increases according to abundance. So the red areas here are those areas, those distributions which have the most cell numbers in there. And this fingerprinting, this is very fast. And you can use this, or we want to establish this, and partly the Rheuma Center in Berlin, for instance, is also using this already. You can use it for diagnostics in poor facilities and hospitals. This is some of the aims. In biotechnology, I'm going to talk about this today, how we use it, and also in ecology, because you can then understand the information on the cells of the communities, the ecological principles that are occurring in these communities. Okay. What do we measure in our communities? So now, somehow, the movie is not running, yeah?
No, it's not going.
I don't know why.
Yeah,
No,
it's going.
not running. Yeah. And what do we measure with flow cytometry? As I said, forward scatter, side scatter, chromosome number per cell. And this is shown here. We have here 4 different strains that are pipetted together. And you see that per strain, we have only subpopulations, populations with different chromosome numbers, which mirror proliferation activity between strains. We also calibrate our data with speeds, as you can see here. And then we create a cell gate, which is shown here. You can see my mouse.
Yeah, we can.
Okay, and so that we check out the noise and the beats, and then we draw a cell gate for each of the subpopulation, subcommunity that is coming up here over time, so that we have a cell gate. And all in all, we measure 200,000 cells, and then we can, and this is the information that we are using for our sample evaluations. And this is, no, here is one missing. So we have, sorry, we have also a mock community that we use to calibrate our data. These are 4 known strains with different subpopulations that you can see here. Yeah, this is here, for instance, the Stenotrophomonas, the Purpurea, here is the E. coli, and here is the Pinnipacillus polymyxa, and the plate mock community has only 3 strains, and they are in certain positions, and then we need this community to align our cytometers to see that the data are in the same place all the time, because later on we use bioinformatics tools to evaluate the data independent of the measurement of the single data. And therefore, we need to be sure that all the data are comparable without any failure. So now I want to start with, now you know the method, and now I want to start what we do want to know on our communities. So first of all, the general definition is a community is a collection of several species living in a particular place. And the members of the community interact both directly and indirectly with each other in the environment. They have structures based on diversity and phenotypic properties, and they are dynamic, and they are subject to both local and regional effects. Local effects are competition and predation, for instance, and regional effects are dispersal. And since we use this meta-community concept, the dispersal between communities for stabilizing, I will come to this. We also need to know what a meta-community is. And meta-communities, a meta-community is linking local community between each other by different dispersal rates. And we have 4 different paradigms how dispersal rates are working, and the one is patch dynamics. So there's little dispersal between communities. There's species sorting, where we have maybe no dispersal. Mass transfer, there is a strong dispersal. For instance, wastewater treatment plants are an example for this. And we have also neutral behavior, where all organisms are equal to each other. This is nothing what is happening in nature, but this is something that very often modelers are using to not have too many constraints for their model. So they just assume that all the bacteria are the same, and they are doing the same thing. So this is the neutral behavior. Okay. So when I'm looking at the community, then I have several things, several events going on in such a community that are responsible for their high variations. And so I have different species here. Where is my mouse? I'm going to apologize. Yeah, so I have many species in here, and when cells are growing in the community, some of them grow slowly and some of them grow fast, and this different growth activities, these different generation times lead to changes in cell abundance of the community all the time. There always is a variation in composition because the species are growing with different velocities. And if I have an open community like here, then species are coming in and species are going out, which also changes the composition of the community. And there are also interactions between cell types, which can be positive or negative. this also changes community composition. And all of this is occurring within minutes to hours. So there can be very fast dynamics in such communities. And since we are going for biotechnological applications, the main idea is to stabilize the community. To do this, we first need to know what a stable community, how can we describe or if a community is stable or not. And for this is we use flow cytometry data for all of this, yeah. So for this is what we are doing is first we determine a reference state. So this is a state which is artificial and is coming from a bunch of samples that we have measured under continuous balanced conditions. And then, because we have always fluctuation in a community due to the cell cycle, we need to create a reference space. And this is about 20% of the fluctuation that we can observe in the community. So, and then if a community due to a disturbance is going out of the reference state, that means that the community is not resistant anymore, like here. Yeah, it's going out of its community. So this is that referred to distance measurement here. And if the community is resilient, then the community is coming back to its original structure after the disturbance. There are also intermediate states, and there is also the possibility that a community is not coming back to its original state. And this is something that we very often observe. So to measure this or to see if communities are stable, we created an experiment where we had 5 different reactors which are completely identically operated and inoculated with the identical inoculum. And we guided this community for 90 days in these 5 reactors. Here, this is a continuous cultivation, so no batch cultivation, continuous cultivation. So always medium is going in, effluent is going out. And the first 2 reactors here were not disturbed, and 3 of the 5 reactors were disturbed by slight temperature changes because we learned a bit that small changes or small disturbances can lead to synchronization in certain conditions. So this is something that we did. So here you see the setup of the 5 reactors, both control reactors, the 3 slightly disturbed reactors, and you see that the inoculum is the same. And now I'm running this, and you can see what is happening over the 90 days. This is a change here of the temperature. And here are the patterns. Yeah, this is forward scatter cell size. This is DAPI concentration. And you see that it's very clear, I think, that 5 different communities are developing over time everywhere. And they are not connected, and they are not similar, they are not synchronized, nothing. And you see this already from the movie, I think. But you can also evaluate this data using the flow cyber. This is a tool that we have developed for our cytometric data. And what you can see here is such a color map. And here on the right side, you have the gates. So we create, as I said before, we create the gate templates of all upcoming subcommunities. We count the numbers of cells per gate. We measure 200,000 cells, as I explained here. And then these cells are distributed, these 200,000 cells are distributed over these gates here. And then you can count them per gate over time, for every day. Yeah, for every day we count them. And then you see that you have dominant subcommunities here everywhere. and you have all the subcommunities that are rare, like here. And the color which you see here is giving you the abundance of cell number per gate. And you see over time that these abundances are changing per gate and over time. And this is this is just here the information for the reactor number 1. And this, of course, can also be observed for all the other reactors. And this is just a precursor dissimilarity analysis, non-metric multidimensional scaling. Here we have the control reactors. These are the disturbed reactors. This is the inoculum. And then and this is the adaptation phases for each of the reactors. So here they are quite synchronized at the start for the first 10 days, yeah. And then they distribute in different directions. So all the communities are different from each other. And also for the disturbed reactors, you don't see a symphony. On the contrary, these communities are even more different from each other. And now we developed some tools to investigate this a bit closer. And one of the tools is, of course, the diversity indices, which we developed here. So alpha diversity is just a richness parameter here for us, giving us the numbers of dominant subcommunities. So the dominant subcommunity is the number of cells that is above the average of cell abundance per gate for all gates. And what we have here, what you see here, these are all the dominant subcommunities for reactor 1 and 2, yeah. And what you can see is that the dominant subcommunities are already different between the two reactors. And they have also different abundances here. So what we can say for this year, this is alpha diversity for reactor 1 and here for the reactor 2, that this alpha diversity is changing. And what is maybe more interesting is that we also determined the intra-community beta diversity. And this gives you the information if you have sudden changes between two samples. So we measure the number of unique dominant subcommunities between two samples over time, just like a moving window, yeah. And then what you see here, this is our reference space here for each of the community. And here we have some events where we have quite huge turnover of community composition. And we name this as stochastic event. So we have a lot of stochasticity here in our communities, which change the composition of the community, which is not caused because we are still in the balanced continuous phase of growth. Normally, nothing should change. Yeah, when you have a pure population, nothing should change. Always the same thing. Nothing is done, nothing happened. Yeah. Nevertheless, we have this huge stochastic variation variations here. And the same you can also observe for the disturbed reactors here in this first one. This looks quite nice already. Here is already some stability in the system, but in the other two, that's getting more worse even because we have here kind of a monodominant community and here also. Nevertheless, after the monodominance, we have an increase in diversity again, in alpha diversity. Yeah, here alpha diversity is going down, going up, going down, going up. And if this is true, because flow cytometry is maybe not so well known, we also took the samples and did a 16S analysis, and then we see here that this 81 days, and here it's 49 days, that we have nearly dominant communities to these time points, and before and after we have again a diverse community. Okay. So we have sudden changes in rank order of subcommunities and a lot of stochastic events here because we also measured here, of course, the intra-community beta diversity. And another tool which we also use is the interaction potential that we have in the community. For this, we need a lot of abiotic parameters, which is shown here. This is the temperature. Then we have the pH, the electrical conductivity. We have measured ammonium, phosphorus, and a bunch of biological parameters. And with the Spearman's correlation coefficient, we can compare the abundance changes per subcommunity with the changes in the abiotic parameters. And here you see the white nodes are standing for the subcommunities with their abundance changes in cell number over time. And the abiotic parameters, which also change their numbers. And then there is a correlation. You have positive and negative correlations. And this is something that we can use to see if certain subcommunities are interactive with other ones or with abiotic factors. And we can also use it to sort certain cell types. If certain cell types have always certain corporations, competition, something running in a community, we can just sort them. And this is what we have found here. There are certain subcommunities here in gray were sorted. We did the 16S, and we see here, for instance, for this subcommunity, this is not changing so much in composition over time. Other subcommunities have totally different compositions. And here we have our monodominant subcommunities consisting out of nearly only one species. You can also take these samples to do the cultivation, sequencing, and proteomics analysis so that you learn a lot about certain species in your community. You can directly take them out and also do this omics analysis. So this is a very strong approach, I think, because we don't have all the background from all the cells. You can just investigate those cells that are interesting for you. Yeah. Okay, and here we have the interaction potential. So this is reactor C from the controls, C2 from the controls, and we have here the drifts, what were happening. Here we show 3 stochastic events for this reactor earlier on, and you see that the temperature here is always, the temperature here is always without any interaction, but this changes when you are going for the disturbed reactors, and always here the temperature is in the midst of the interactions. Yeah, you see that here indeed the temperature does something to the cells in the reactor system, but always different cells responded to the temperature. It was not always the same. It was always a different one. So therefore, we concluded that we had still a lot of stochasticity here in this system. And just to conclude with this first approach that we tried to synchronize our communities, to stabilize our communities. So this was obviously not successful. So we were not successful doing this. We had a different structure and a different evolution. We found no synchronization and stabilization, no resilience, no resistance. Stochastic events were even higher in undisturbed reactors. And the interaction types between the dominant subcommunities was always changing. So there was nothing we can use to go on, to get ideas to go on. And the interactions even increased in disturbed reactors, but always between different subcommunities and the temperature. So what was going on here, how we can describe this, what theory is behind this. And this is the intermediate disturbance hypothesis. And the IDH says that intermediate levels of disturbances can increase and sustain diversity. This is actually what we saw, yeah, for our alpha diversity values here. So there was at least a stability, even a small increase here. And this is because which type of disturbances we had in our balanced systems, balanced continuous systems. This is first is the dilution rate, and second it's drift. And we lose also the cells out of the reactors. So we always lose cells out of the reactors. So because of the dilution rate and the loss of cells and the drifts that are occurring in our community, there was no establishment of monodominant communities, which also some predict over long time ranges that this is happening. And therefore, the alpha diversity remains high. And in addition, I show you that we had a lot of rare cells in the system. And if we lose organisms due to the effluent, there are free niches for the nutrients that are coming into the system, and this can be overtaken by the cells that are in the system that are the rare ones. And since we use the wastewater community as a starting community, you can imagine that many of these organisms in the system have functional equality, and therefore all of them have similar chances to survive or to overtake even dominant situations or dominant positions in a community over time. Okay, so what do now? We were looking in macroecology. And you always have systems which are stable over a long time. Now, when you're going to the woods, you have small spaces and meadows in between and so on. And this is stable over time. And why is this the case? And why do we have the same plants on different meadows that are in a regional scale where we are looking at? And this is dispersal. And now we are coming to the meta-community concept. So dispersal is a mechanism which keeps larger systems stable. Yeah, if an organism is lost, for instance, I'm always telling the story. Yeah, when you have in one wood a fox, and this fox is shot by a hunter, then and in another wood nearby you have a fox family. Then what would happen? One of those foxes will move into the empty fox, yeah, and sweep over there. And this is dispersal. And the same thing is what we wanted to use here for our bioreactor systems, to create a stable environment. And this is the composition. We have here the 5 reactors. The same reactors, identical reactors as before. But now they are connected, each of the effluents here is connected to a regional pool. Another reactor, 6th reactor. And from the 6th reactor, the cells are going back into each of the other reactors, as you see here. Here we have the medium inflow, and here we have the effluent. And yeah. And what we were doing now is we establish continuous systems. We have the balanced phases, nothing is changing. We inoculated with the same inoculum all the reactors, not the regional pool. And we let the reactors run without connection and with connection, and with connection with different recycling rates. So part of the inflow was then exchanged by the inflow from the regional pool. Nevertheless, the medium concentration was always the same because we increased the medium components according to the recycling rates. And this is what happened. So now we have the insular phases. Reactors were disconnected, and you see as before, different communities are involved. And now we start the recycling rates, 10%, 50%, and 80%. And you see that we have now stable communities and synchronized communities. We have the communities are all in the same composition. And this is an insular phase again, meaning that the communities are disparate. Excellent. Should I run it again? Okay. We can also show these outcomes with NMBS plots, yeah, and you see here the insular phase with the different colors are standing for the reactors. Here is the inoculum. This is the adaptation phase. And then they are going all in different directions. Yeah, and then you have RC10, RC50, RC80, and you see that the community is getting more and more similar, stable, and all the communities are getting synchronized. And if you disrupt this, then they are getting again in different directions. Okay, and then of course we can use different tools again to interpret this data. And in this case, we have used inter-community beta diversity between the 6 reactors. And this is shown here. So we compared pairwise between reactors per day for the local reactors and also the local reactors against the regional pool. And what you can see is that the lowest diversity is under RC80, it's here. And it's going up, the diversity is going up again in the insular phase 2. When we measure constancy and resistance, then we see that we have the lowest constancy spaces under RC50 and RC80. Also the lowest fluctuations in the balance situations of the communities. And for the resistance is highest also in the from the change from RC50 to RC80. And in addition to this, we measured also beta diversity partitioning, meaning that we wanted to know, do we have nested sub-communities in the system? Meaning that we have sub-communities that are always there during the whole time point. And do we have, and how much is the turnover, the exchange of sub-communities, yeah? And then you see that the nestedness is fairly, not changing, but the turnover is going down a lot. Yeah, we have a lot of turnover in the insular phases, as you can see here, communities exchanging, but in the under RC80, the turnover is quite low. And we also use tools from other groups, not only our own. So we use the QPAN from Stegen, which is a tool needing sequencing data. So we also sequenced our all these samples, and all of these. So we had for the sequencing less samples, the 16S. But also with this tool, also based on Friedrich comparison and for determining the homogenization, homogenizing dispersal, and we also see here for the Stegen tool that we have the highest homogenization dispersal under RC50 and RC80, and the NST is a tool from Ning, who is determining the degree of stochasticity based on a model where he assumes that everything is stochastic. And this is also the lowest stochastic events, or the lowest amount of stochastic events is occurring under RC80 here. Yeah, and this is a slide where we determined the net growth rate of the subcommunities. So you remember we have about 60 subcommunities per sample, and these are the gates here, and these are the insular phases here, the 1 and 2, and these are the recycling phases from 10, 50 to 80, and each point gives you in red, and the height of the red, the degree of net growth. Yeah, so we can determine for each of the sub-community how well it is growing in the community. And what you see here is that we have in the insular phases a lot of dominant sub-communities that are there first and that are growing, right? And this is because between the different between the different reactors, of course, I have very different sub-communities surviving or be dominantly present in these reactors, but different ones. But as soon as I start the loop design, then the number of dominant sub-communities is going down. And also, you see it for the color, also the growth rate. And this is because due to mass transfer, my biomass is increasing threefold, yeah. And yeah, threefold growing. And then I have less and less sub-communities that are growing in the system. And less sub-communities and less that are growing in the system. And this is kind of counterintuitive maybe, but the sub-communities are staying in the system because I have an exchange. Yeah, and so I have the regional pool, and if the sub-communities are growing in one of the reactors, they will always distribute it via the regional pool back into all the other ones. And therefore, these sub-communities are staying in the system. And ecologically, in the meta-community theory, this is called the rescue effect. Yeah. So I have coming from the dominant sub-communities percentage over 1.25% over all sub-communities that I have in the system, with a net growth rate over 0, larger than 0. I have 305 when I start, and only 23 are surviving and are present in the system for a long time, finally. And but they grow more slowly. And this is due to that I keep the nutrient composition, the carbon energy source stable in the system. I do not increase this. I just concentrate it to equalize the recycling rate from the regional pool. And then due to the rescue effect, they stay in the system for a long time. Okay, so now I'm coming already to the summary. And so what we found, first, flow cytometry is a very fast tool to see this. Yeah, this is we can measure, as I said, this stuff in a few minutes, and evaluate in the same way, very easy. The tools are very easy to use, and you have the data available in an hour or something. So what we saw is that when we were using the loop design, microbial communities can be synchronized. Yeah, you just don't need only one reactor, you need several ones, and you need to connect them via regional pool. And the rescue effect is the main effect that is active here, and which is supporting the synchronization of the communities. Yeah, important applications are the health sector. Maybe you heard about this continuous fecal transplantation. Yeah, if someone has an inflammatory bowel disease, then this can be healed not just by antibiotics, which is very painful and not always working. But if you have fecal transplantation, you need to take it all the time. Yeah, this is like a leukocyte reactor, what we are having in the gut system. Yeah, and then you may also know that certain animals, like chicken, for instance, eat their own feces, and this is also somehow stabilizing their own microbiome. Yeah. In biotechnology, there are not so many applications right now because the companies do not use these leukocyte till now. Of course, it's also new that we developed this. Because they have the trouble that the communities are always changing and that the systems are not stable. And we believe that the look design can be a way to change this. And of course, you can ask a lot of ecological questions with these types of experiments. And we have a lot of ideas which we need to follow, which we want to follow also here. Many unanswered questions and which we can use. We have developed a lot of biostatistics tools based on R. They're relatively simple, but work fine. So we can do we have the flow check, which gives you an immediate overview on community evolution. And with the flow cyber, you can go more deep, you can go per sub-community, and you can do the similarity analysis, the interaction potential, the different diversity values, turnover nestedness, and stochasticity values can be determined. And of course, the stability measures and the net growth rates. And this is always in the frame of developing these communities. I think you are also inside of these concepts, yeah, so that we can go bottom up using certain species putting together. Yeah, when you go bottom up, you can even use genetically engineered organisms which have a certain function. But these organisms are not used to each other, so maybe there are problems. But you can also top down reduce the diversity of these communities to not have a lot of species eating up all the nutrients. Yeah, and you can go with a reduced community for product production. And with this, I would like to end my talk. And yeah, they can tell your questions.
Thank you, Susan, for the nice presentation. So we have some minutes for the discussion. So I would like the audience to raise the hand or just open the microphone. Yeah, I see already Caroline. Please, you can go.
Yeah, nice to listen to this very interesting presentation and to see you again, even if it's only online. Yes, I always follow with great interest what you're doing. I had a very fundamental question on the trouble with the heterogeneity, the phenotypic heterogeneity. So how much of this change in gates is due to different taxa and what may be due to simply species doing different things in the presence of other species?
You mean how we said you mean
the gate definition. The gate definition assumes that the species are stable, but they may do different things and look different in gates when there are other species present because they adapt.
What we are doing is that we set about 60 gates. Yeah, and of course, when you have a natural community, you have much more species in the system, maybe 500 different species. And that is naturally that you have per gate different species or different types of cells. But what we know from pure populations, and this is something that Rosalind we also confirm, yeah, then these pure populations only occur in certain positions of the histogram. That means that they go into certain gates only. They are not present in all the gates. They are just present maybe in 5 different gates. And when they change gates, that means that they are growing. Yeah, so they are going from the lower gate to the higher gate when they proliferate the DNA. And that means that they grow, meaning that they are active. right? And this is something that we are measuring because we measure number of cells per gate. And when the number of cells per gate is changing, this gives you an indication that something is going on in the system. And this is also something that you see then in these interaction maps. Yeah, when the cell number per gate is increasing due to proliferation, what we measure is actually proliferation growth. And this is the most active cell parameter that we can measure growth. And this is what DAPI and forward scatter, also the forward scatter is changing when cells are growing, it's getting larger. Yeah, for instance, or when you have spores in the system, this is also when there is an additional gate which is filling up the spores, which is increasing in cell number. And these change is not a sequence, but it's a kind of physiological information, which nevertheless, and we always, because we know this concern of the reviewers always, we always do the sequencing analysis, 6S analysis in this, and it shows us here, but the trends are totally identical. Yeah, so this is something that we can, it's going in the same way. So it's a very fast method. It's going for physiological parameters, and the differentiation is less. It's maybe also, it's like on the genus level, you have also not more than 60 different genera in a sample, right? When you're going for a natural community.
Yeah, you don't need to convince me of the value of the method. I was rather asking about the phenotypic heterogeneity, how you could exploit it. But I think it's even more powerful, I think, actually, than sequencing, because you see the different phenotype superpopulations that you may not so easily see with sequencing. So yeah, I was just wondering if, but it could maybe be, yeah, anyway, so we can discuss another time. Thank you.
Thank you. Rosalind, please.
Yeah, thanks for a really nice talk. I was wondering, maybe I missed it, but what is really a sub-community? Is it? I understand that it's one of the gates in the flow cytometry, but is it like one species, or is it like a little collection of species?
So as I said, we have the gate template. We have the gate itself. Yeah, this is a certain space that we have available to differentiate cell types from each other, and we measure cell size against chromosome number. Many cell types, also from different species, have the same distribution.
Okay.
Differentiate the cells according to Gaussian distributions. We set the gates in the valleys around the Gaussian distributions. And these gates templates we create over several of the histograms, and then we use this for everything that we measured. Yeah, and these cells in one gate, so we have 60 gates, and these cells in one gate can be, as I said, monodominant, we call it, when I have nearly if only just one species type is in there due to ecological situations, as I showed you, or there are very different cell types in there. And this can also even change over time. Also, the composition of the, when we are going for long time ranges, yeah, then the composition can even change. And therefore we need from time to time also to sort to see which cell types we have in there. But when you are going for the dynamics and see how the dynamics are going, and to see that nothing is changing, then you can pretty sure assume that you have always the same species type in that per gate. Yeah, but if it's changing, then it's sometimes worthwhile to go into the gates and see what else in there, what happened. Yeah.
I'll ask another quick question. Sorry, Andreas. If you had a system which was making kind of aggregates, so like, you know, some species were transiently aggregating together, could you check that by flow cytometry if you didn't like disperse the cells, but you just kind of
this is a question which is also always coming. Flow cytometry is in the first place a single cell measurement method. Yeah, it's like also for medical applications. This is developed for measurement, for the measurement of the single cell. And what we do is we wash our cells in buffer, and this dissolves most of the aggregates. We have nearly no problems with wastewater aggregates. But of course, when you are going for I don't know, species that stick together very hard, yeah, which even construct a cell wall between each other, yeah, or over all the cells. This is something that we cannot measure. So we sieve our cells also. So part of the cells are lost. But I need to say that we, due to this question, always coming also from reviewers, we also do always, as a proof, sequencing, sequencing. We go through every step and see if we lose some cells at some treatment point, and what we see is that the differences are nearly not seen. Maybe we lose some cells, but the majority, really 95% of the cells pop up in the flow cytometer, and this is proven by additional or by parallel sequencing on the genome level.
Okay, thanks.
Mm.
Yeah, I think we're running out of time, but Andreas, please. Yeah.
Maybe a quick one. I was wondering about stochastic events. So there can be maybe gene expression differences. There could also be like mechanical differences or like temporary variants of the nutrients. Do you have any assumptions? What could be behind that?
Temporary variants of the nutrients I would exclude. At least on the reactor scale, how it is between cell types is maybe we cannot measure this, yeah. But we have continuous reactor systems in balanced spaces. So normally, when you have a pure population, you can grow them over years. And this is besides if there is a mutation, of course, and something might happen, yeah. But this is really very stable. Noise, of course, when you have a pure population, you can follow transcriptional noise. This is, of course, difficult in communities to follow, can play a role. But I think what is happening here is more the ecological part. This is competition between cells that have the same function. This is functional redundancy. So cells are compete with each other on the nutrients that are available. The nutrients are set, so there's the same condition all the time, so they compete. And sometimes the one wins and the other wins, maybe how they are interacting with in this moment can lead to these decisions, but this is random, yeah. And so I believe, or I think, what we observe, this is real ecological stochasticity what we are seeing coming from competition, cooperation events.
Okay, thank you very much.
Thank you. We have one question in the chat, and after I will go to Tian. Okay, the question in the chat is, okay, thank you for the great talk. A technical question. How do you keep anoxic conditions for microbiome analysis?
This what I showed you here today is done under oxic conditions. This is the wastewater community. We have also done this, not the loop design, but flow cytometry on digesters. So we just use digesters, so we don't need to keep anything there. And we have a cooperation also with the Biomass Center here in Leipzig, where we have had running anaerobic digesters, so they have a lot of experience, and it just takes longer, it's more complicated, but the flow cytometry is the same.
Thank you. This one was a question from Sivan. I hope the pronunciation is correct. Now is Tian, please.
Hi, Professor. Thanks for your talk. I'm a PhD student from KU Leuven. I'm working on individual-based modeling, and so I want to ask a question that because that many bacterial species, they are living in biofilm settings, that they have EPS matrix, that they have local environment. And so how the special structure that they formed that you think will affect the system properties on the different temperatures or different environment perturbations? Thank you.
How species react to different perturbations? Is this your question?
Yeah. So because that they will have like they are living in biofilm, they have EPS matrix, and they
The biofilm. In the biofilm. Okay. Okay, biofilm is a completely different topic. It's also not so easily measured by flow cytometry. Yeah, Rosalind, you have this issue. Yeah. Yeah, you need to be creative to get the biofilm from each other. Yeah, I mean, biofilms are fascinating, and you have very different types of bacteria in different locations of the biofilm. Also, if you talk about pure biofilms, we know that we have in every position that we can do a biopsy in the biofilm. We have such a paper published recently that you can take biopsies of different positions in a biofilm, and then we see that cells are the composition of the population is completely different at different locations in the biofilm. Yeah, so for instance, for spore producers, we find most of the spores in the inner ring of the biofilm. And also cells that produce toxins, for instance, we also found in the inner ring of the biofilm. Yeah, we can measure them, we can quantify them, and can connect them with the toxin production in a certain location in the biofilm. So this is flow cytometry can do this also for pure populations, which is nice because we can do nothing here with sequencing. Yeah. Then you need to go for the transcriptomics or for the proteomics to know more of the functions. But for instance, for Bacillus subtilis, we can differentiate such a population into 12 different subpopulations. All of them have a different physiological state. And this is really the beauty of flow cytometry also when you are going through this. But we have also biofilms. I need to say we tested different types. We have also biofilms which are not as structured, where we have only 2 different cell types or so. Yeah. So this is really dependent on the species, on the growth conditions, and everything. But
yeah. So I think that the individual-based modeling can be used to derive some hypotheses, because I saw the Professor Volker Grimm that also in your acknowledgement slide that he's the leading scientist for individual-based modeling. Thank you. Thank you.
It was a great help actually. This was really fun to work with him together here. Yeah.
Thank you.
Thank you. If there are no other questions, actually we are running out of time. I will